So, like I said, I'd like to thank the organizers for the invitation to come tell you about my work. So my interest is a bit different from many of yours. We're using deep mutational scanning to look at questions in trying to understand protein evolution. And so we're interested in what do real fitness landscapes look like, what causes the fitness effects of mutations. So I think there's enough overlap with your interests that hopefully that you will find something that you find something interesting in what I have to tell you today. So for that, because we're interested in protein evolution, we do our work. We work exclusively in E. coli for simplicity's sake, and so what our readout is fitness, and by fitness I mean growth rate of the bacteria. So our typical experiment, we would make our library of a comprehensive site saturation mutagenesis library in E. coli in our gene. We would sequence that. We would start. We would. Okay, the pointer's not working. We would. It's still not working. I am not touching that button. So what we do is we start our bacterial, the gene that we're interested in is on a plasmid. It is under the control of an inducible promoter, so we're not expressing. We grow the cells exponentially. Then at time 0, we start expression of the protein. We take a sample, we do deep mutational scanning, we sequence that. We grow the cells for 10 generations, and then we deep sequence. Thank you. Then we deep sequence and determine the frequency of mutations after 10 generations. And then based on that, we can tell the growth rate of how each mutation affects the growth rate of the bacteria. So one of the genes that we use, just as an example, is TEM1 beta-lactamase. So for those of you that have ampicillin resistance on your plasmids, that's TEM1 beta-lactamase. So it confers resistance to beta-lactam antibiotics. It's exported. It's an exported protein. It's a convenient model protein for studying protein evolution. So when we're studying TEM1 beta-lactamase, since its only known physiological function is to provide resistance to beta-lactam antibiotics, we do an experiment where, we grow the bacteria in the presence of ampicillin. And so, a result of the experiment like that might look something like this. Now, this experiment was done different than that. The details are not important for this talk. And I just want to point out that what we have, our fitness scale is quite severe. So even things that are growing 50% slower are full red here. And when you look at this landscape, you're tempted to look and try to understand things that are happening. You can see things like here in the signal sequence, which is important for export. You can see that charged residues are not tolerated in this hydrophobic core, and that's well known from the literature on TEM1 beta-lactamase that that's the case. So if we're interested in what's the relationship between the protein properties and biological fitness. And so for ampicillin, you might write down this sort of simple equation, that fitness is some function of the total catalytic activity in the cell, since beta-lactamase's only function is to degrade beta-lactam antibiotics, and the amount of ampicillin that's present in the environment. And that total catalytic activity is going to be a function of the specific activity of the protein and the protein abundance. So your mutation might affect protein abundance, might affect specific activity, might affect both. And for what I want to argue today is that that is a very simplistic. It's ignoring a lot of what might be going on in the cell. So that's because the cell is very complex. So not only must beta-lactamase carry out its physiological function, but if it has a mutation, that mutation not only shouldn't harm the physiological function, but also can't interfere with anything else that's going on in the cell. And so if the mutation causes the protein to aggregate, if the mutation causes a misinteraction with some other protein, if the protein might aggregate with another protein, might, because it's aggregating, cause chaperones to be titrated out, and that has a growth effect, or might induce a stress response, and induction of that stress response causes an effect. So what we've proposed is that it's useful to delineate between primary fitness effects, those effects that arise from the ability of the gene to perform its physiological function, and collateral fitness effects, which are those effects that arise from factors other than changes in the ability of the gene to perform its physiological function. And so one of the good things about using antibiotic resistance genes for this is because those antibiotic resistance genes, that at least the ones that we've chosen, have only one known physiological function for the bacteria, that is to provide that antibiotic resistance, at least for the genes that we've chosen, that we can ask the question, how often are there these collateral fitness effects? Simply by repeating our experiment, but in the absence of its cognate antibiotic. So I want to point out, though, that we have a secondary antibiotic resistance on our plasmid to maintain the plasmid. And so we did that experiment on these 4 genes. TEM1 beta-lactamase, a different beta-lactamase NDM1, chloramphenicol resistance gene, an aminoglycoside gene that confers resistance to aminoglycosides. And you can see that so I apologize, I've changed the coloring scale here. What we're showing is where do we see a statistically different growth effect from wild type, and we only see deleterious effects in all these experiments. So mild deleterious effects are here, from a 1% decrease in growth rate down to a greater than 30% decrease in growth rate. Okay. So what I want to point out here is that these effects, especially with that TEM1 beta-lactamase, are not because we're grossly overexpressing our protein. We can see here's beta-lactamase under our inducible promoter. Here it is under its native promoter. It's slightly higher expressed, but not by a factor of 2 or 3-fold. Importantly, whether you induce expression of the beta-lactamase or not, the growth rate of bacteria over 10 generations is the same. We can't basically tell the difference statistically. And if we look at gene expression here, yes, when we induce with the IPDG, we see induction of the LAP operon, but we see very little else happening. And if we compare the plasmid where there's no beta-lactamase gene with 1, there's basically no difference. So the cell is not really feeling the effects of expressing this beta-lactamase, at least to the extent that we can measure. Yet introducing mutations, over 42% of the mutations in TEM1 beta-lactamase are deleterious. So this slide here is just to remind me that we do validation on this, on select mutations that we want to study, where we measure growth rates of monocultures of cells expressing with these mutations. And when you have neutral mutations, they're neutral. And when you have deleterious, they're deleterious. So let's look a little bit at what's happening in TEM1 beta-lactamase. You can see that there's patterns of where the mutation, and I'm back here to the other scale, where red is deleterious here. And we're only showing mutational effects where it's deleterious in 2 replicas with a value of 0.01 in both replicates, p value. So we can see that there's mutations in the end of the signal sequence. There's cysteine is the most deleterious mutation, mutations to cysteine, and that in the center of the protein, it's this alpha domain here. There's a large amount of protein, a large amount of mutations are deleterious there, and that part of the protein is known to fold first. And so you might expect that this has something to do about reduced stability of the protein or reduced folding rates, perhaps causing aggregation. And if you thought that, you would be right. So we looked at what's happening to the mutations. So type of mutation signal sequence. What they cause is diminished or improper cleavage of the signal sequence and aggregation. They don't cause the export pathway to be compromised, surprisingly, which is what I thought we would find a lot of. It just interferes with the ability for the signal sequence to be cleaved off. The signal sequence is known to retard the folding of the protein, and you end up with aggregation. The cysteine mutations, we get some incorrect disulfides where two beta-lactamases form disulfide bonds between them, two beta-lactamase molecules, and we also get aggregation. And then those mutations in the center part of the protein in the alpha domain also cause aggregation. So aggregation, aggregation, aggregation suggests that aggregation has something to do with these collateral fitness effects. This is just a quick aside to say that sites that cause deleterious collateral fitness effects tend to also cause deleterious primary fitness effects. But there are sites that have no primary fitness effects but have deleterious collateral fitness effects. So there are mutations. So this is if your readout is biological and your interpretation is something about the protein, this is a word of caution. Okay. So since it looks like aggregation has something to do with these collateral fitness effects, we wanted to look at this sort of systematically to sort of get some more evidence of aggregation's importance. And so if you're trying to express a particular protein and overexpress it, and you're having problems because it's aggregating, one common thing you do is reduce the growth temperature. When you reduce the growth temperature, you're reducing the rate of protein synthesis, you reduce the concentration of folding intermediates that tend to aggregate, and so you get less. And so you get less aggregation. So we thought, well, if we do the experiment where we reduce the growth temperature to 30 degrees, we would expect to see, if aggregation is important, we expect to see a reduction in the frequency and magnitude of the deleterious collateral fitness effects. And that's indeed what we see. Another way that you could do, but it's growth at a different temperature, it's in a different environment. So we decided, well, what if we keep the temperature the same, but we change the media to be a minimal media? That will also slow down the growth rate. Would that also have the same effect? And that also had the same effect. So really, it's pointing to aggregation. If you raise the growth temperature, now aggregation tends to be more detrimental. However, raising the growth temperature above 37 in E. coli causes induction of stress response, which goal is to ameliorate those effects. And so when we do that, we kind of get something that's a little bit redder, but not too much. What's interesting is if we take the difference now, we take the 37 degree fitness minus the 42 degrees fitness, and we look at that delta landscape here, we see that although there are, and these are all the ones that are statistically significant, although there are certain positions where it becomes much more deleterious at 42, there are also positions where it becomes less deleterious at 42. And so we wondered what was going on there. And we also wanted to see, okay, now that we change the growth conditions and we see less collateral fitness effects, do we also see less aggregation? And the results there are very mixed. So if we look at some mutations that get much more deleterious at 42, we see in some cases that we get a lot more aggregation, and in other cases we get less, although we get aggregation of something where the signal sequence is not cleaving. When we look at things where the deleterious collateral fitness effects get worse and then get better at 42, we can see there's usually very little difference between the amount of aggregates at 30, 37, and 42, but it does decrease at 42. And a similar story here, that sort of points out that maybe it would be very satisfying if the amount of deleterious collateral fitness effects was proportional to the amount of aggregation, and that's not the case. And to sort of hammer home that, here's looking at a couple other genes where we see that you could have lots of aggregation and no deleterious collateral fitness effects. You could have mutations at the same position, very conservative mutations. One is completely neutral as far as collateral fitness effects. The other is very deleterious, and both of them seem to cause the same amount of aggregation. So what I would say is that what is likely causing deleterious collateral fitness effects and what causes aggregation is probably something to do with destabilizing the protein, but it's not necessarily the aggregate itself that's the toxic species. It might probably be something that the protein might be misinteracting with something else. So our hypothesis is that perhaps the deleterious collateral fitness effects arise from misinteractions, right? So the aggregates couldn't be toxic on their own. It has to interact with something, otherwise it can't have an effect, right? And so if that's true, we would expect that different proteins and differentially mutated proteins should misinteract with different partners. And if that's the case, maybe the cell would respond in different ways to those effects. And so we next asked, what does the cell think is going on? So we did some RNA-seq experiments here. And by and large, what we found is mutations that cause, the greater the deleterious collateral fitness effect, in general, the greater the transcriptome is perturbed. And there's some general things that happen that make sense. So if we look here so this is a particular stress response pathway in E. coli, the phage shock protein response, that in our secreted proteins, so it's a response to intermembrane stress, and our secreted proteins all mutations in there seem to induce that, but our cytoplasmic proteins do not. That seems to be satisfying. However, there's an outer membrane stress response here that typically is known to be activated in response to antibiotics. And we can see again that our periplasmic expressed proteins do that. However, none of the signal sequence mutations that are deleterious activate this response. And we have cytoplasmic proteins that activate this outer membrane stress response. And we even have mutations at the same position, ones that will upregulate some pathways and downregulate other pathways. So this is all to say that this is much more complex than just aggregation. And oh, well, I actually have two more examples in the last minute here. That these deleterious mutations that cause deleterious collateral fitness effects can have some very unusual phenotypes, which again suggests there's something very specific going on, that these mutations are causing the proteins to misinteract with specific other proteins. So one of them is we found that there's certain mutations near the beginning of the chloramphenicol resistance gene, that mutations to methionine were deleterious. And we basically could show that what we're getting is internal translation initiation. And what was interesting is there's a single copy episome in this bacteria. Those mutations, these two mutations here to methionine, cause loss of the episome. But similarly deleterious mutations in the same protein, even though they're similarly deleterious, do not cause loss of that episome. So something specific is happening there. And an even sort of more bizarre phenotype is there's certain mutations in this protein here that cause this very unusual antibiotic resistance phenotype, where if you particularly look at this mutation causes the bacteria to require intermediate levels of the antibiotic to grow. If the antibiotic is too high, it doesn't grow. If the antibiotic is too low, it doesn't grow. Anyway, again, it points to something very specific and interesting is going on. So I'm out of time, so I'll just skip the conclusions and put up my acknowledgement slide to thank those who did all the work. Thank you for your attention.
Thank you. You did very well on time. So we have plenty of time for some questions.
Oh, that was 20. I thought it was started at 30. That's why I rushed.
You can go back to your conclusions if you want. But we could start with questions and, as you wish. But I think there was a question back there.
Hi, thank you for the talk. Super interesting. So when you sort of present this range of single fitness values that you've sort of calculated, I assume, by sort of measuring to like your relative controls, how do you compute a single fitness value? Do you sort of, you say you take this measurement over like 10 generations of growth. What maybe analysis or what computation do you do to back solve a single fitness value?
So it's basically, you do deep sequencing, you have a frequency of that allele at generation 0, you have the frequency at generation 10, you also have the frequency of wild type at those positions, and therefore you can back out the relative growth rate because you know how many generations there are. So it's like if some of you do things where like some enrichment score, enrichment factor or enrichment scores, it's something like that, but we're also including growth rates, so we're actually getting relative growth rates there. And
so do you ever sort of look at like growth measurements of a culture and maybe like create a value that maps back to that, or do you always use sort of the sequencing-based measurement of fitness?
So we use the sequence to get the whole landscape. We use the sequencing base, and then we pick particular interesting mutations and validate them by measuring growth rates relative to wild type in monoculture.
Okay, thank you.
So while we're looking for the next question, so you drop this intriguing, like you need, this intermediate amount of antibiotics to grow well. So you kind of drop that at the end. And can you speculate a little bit about what might be going on there? Like I know there was a paper way back when of, bacteria that are evolved to eat antibiotics as their carbon source. Like, is that what's happening here?
No, I mean, no, because they're growing in rich media here. So what I think is probably happening is so the mutation causes destabilization and misinteraction or some deleterious effect, all right? When you add the antibiotic, when the antibiotic is, the protein has to bind the antibiotic, that's going to stabilize it, all right? So, so you have to add enough of the antibiotic
to keep it busy.
So when you add enough of the antibiotic, then the protein is not going off and doing what it shouldn't do because it's always being occupied by hydrolyzing the antibiotic. But then, of course, when you get too high in antibiotic, there's not enough catalytic power to deal with that. So that's what I think is going on. And we have some experiments with the beta-lactamase. We started to do some landscapes where we do TEM1 landscapes in the presence of beta-lactamase inhibitors. And that's kind of, we just have preliminary data, but that's sort of bearing out. If you add a beta-lactamase inhibitor, it decreases the deleterious effects of the deleterious collateral fitness effects.
Okay, here we go, good, yeah.
Hi, thanks for that really interesting talk. Matt Dale from Weill Cornell. I just wondered if you thought that the mutations to cysteine were surprising in an E. coli setting, given that the cytosol is reducing.
So TEM1 is secreted to the periplasm where disulfides form.
So do you think the aggregations for those mutations are specifically in the periplasm space? And does that matter where the aggregation happens?
Yes. We did experiments that, one, the disulfides are forming, so that suggests it's in the periplasm. And we did experiments where we blocked the formation of disulfides before we lysed the cells to verify it wasn't happening after the fact, after we lysed the cells. And the signal sequence is cleaved off, which also suggests it's secreted.
Okay, we're trying to get the microphone around you. Great, thanks.
Hi. So the fitness effect is presumably a function of the expression level that you have and of the selection pressure. So have you been able to vary either of those two and see if the collateral fitness changes as a consequence of those variations?
Well, as far as the selection pressure, what would be the
The
selection?
antibiotic concentration.
But we don't add antibiotic.
Okay.
So we don't add antibiotic. Yeah, so the selective.
No, but you're looking at the difference between adding antibiotic and not, right? Or
No, we're looking at what is the effect in the absence of antibiotic.
okay.
But the first part of your question, what was it?
The expression level.
The expression level.
Because
we
the
haven't looked-
aggregation will depend on, or whether you get proteolysis, aggregation,
Yeah,
whatever.
we haven't done that experiment. I mean, I think it's, you expect as you increase the expression, the effect is going to be worse. If you decrease the expression, it has to eventually go to zero. Where the what exactly the shape of that is, we don't know. We did have one example where, once we want to validate these mutational effects, we have to make that mutation. And one time we made the mutation, and suddenly it was had such a huge deleterious effect. It was making a ton of protein, and we figured out that there was that the plasmid had acquired a mutation that affected the copy number, and that was why it was. So that, with one mutation, that addresses that question.
Okay, I think we have one more, and then we'll move on. Sorry, over here.
Oh, where? Sorry. I was looking up. I'm sorry.
Great talk, thanks. Joe Marsh, University of Edinburgh. People are often doing MAVs of proteins in non-native contexts. So I wonder to what extent you think these kinds of issues might be pervasive in influencing the fitness measurements that have nothing to do with perhaps the biological context of the proteins.
Yeah, so that was one thing we were, in trying to figure out, what proteins we wanted to use for that study. We wanted to make this as native as possible. So TEM1 beta-lactamase was originally discovered in E. coli, but it's a plasmid-borne thing. It's not a part of the E. coli genome. So yeah, we chose to try to be as native-like as possible. But I would think, you just have if you're expressing under non-native conditions or in a heterologous host or things like this, you'd think there'd be even more opportunities for this to happen to begin with, just because without any mutation, that expression of that protein is causing some effects. So yeah, but the other thing I would say is, I didn't point this out, but the magnitude of the effect of these are for most of the mutations are very small. So these were most of them are 1 to 5% decreases in growth rate. Now, evolutionarily, that's huge. You have fitness effects of way less than 0.001% things like that are going to be wiped out by evolution if you have a fitness effect of that effect. So, but we can't measure things down that low. So the effects are generally pretty small, and the ones that have very large effects are rare. So hopefully they'd be rare and not, but it's something to keep in mind, I would say.
Great, thank you very much.